Hello, my name is Jhneal Williams and I'm doing MC1R for my protein project. So, the location of this protein. This protein has 317 amino acids and it is located on chromosome 16. It tends to be found on the cell membrane in cells that possess melanocytes. Essentially, it tends to be found in mammals in comparison to like plants. So the function of this protein is to regulate pigmentation, and it switches pigment production from pheomelanin to eumelanin. MC1R protein is characterized as a receptor that binds to signaling molecules to regulate melanin production. So the mutation that we are dealing with, this is a single nucleotide substitution, so there's not anything changing except one single nucleotide, and this is specifically on amino acid 67 where arginine changes to cytosine. So the single change in the amino acid sequence blocks and prevents the ability to bind a AMSH, which interferes with melanin production, and what this causes essentially is decrease eumelanin, which is the dark pigmentation, and increases the phenomelanin, which is associated with fair skin, red hair, and increased UV sensitivity, and this can further lead to the condition of skin cancer. So this is like the phenotypic breakdown of what would happen. So, the red and orange pigmentation in the hair, and yeah, that's basically that. So with the amino acid sequence, originally we have like a positively charged polar amino acid, and it forms hydrogen bonds and ionic interactions. Additionally, it helps stabilize MC1R's extracellular structure. The main interaction is the protein-protein interaction that causes the receptor to bind and start the signaling. The mutated amino acid is now nonpolar and forms disulfide bonds, which may disrupt the charge interactions and receptor conformation. The interactions that is possessed in this mutation is hydrogen bonding at a much weaker extent, and the ability to form strong ionic interactions with negatively charged molecules is lost due to this substitution. So here I have the structure of MC1R. So we have this 3D form, how it looks when it's bundled up, and this is like a basic breakdown of the structure. So basically, MC1R has 7 transmembrane A helices, which makes it the compact globular shape that I showed on the right side. And these helices span the entire plasma membrane, and intracellular regions interact with the G proteins, which initiate the signaling. So in terms of the degrees and the structural components, the primary structure is the linear sequence of amino acids. The secondary structure is those A helices between the transmembrane that I was discussing. And the tertiary structure is a 3D fold that folds the plasma membrane into the globular shape. However, MC1R does not have, it functions as a monomer, so it does not form multi-subunit complexes. So with my model, I have right here, not very sturdy, but so on this side we have the amino acids starting outside of the transmembrane. The green popsicle sticks that I'm holding represents the membrane. This would be the extracellular, the bottom would be the intercellular, and the 7 green double-stranded helices are what are in between. So the pipe cleaners, as I had stated, were the A helices, and the foam balls are more for like stability and the extracellular regions, and then the beads are the amino acids. So basically what I have here, adenine is the light pink, thymine is the light purple, guanine is the dark pink, and cytosine is the dark purple. So with this, if I were to cut at the 67th amino acid, let's say for instance, one of these, it would have to be one of the light pink balls, and I would replace it with a dark pink, and that would cause the mutation of this membrane. It would still look the same essentially, but it would not function the same. But this is my protein model. And essentially, like I said, cutting at that 67th amino acid, replacing the light pink with a dark pink, which is what I would say is the adenosine with the cytosine. So the limitations of my model, obviously, it's not that sturdy. The actual receptor undergoes conformational changes, which is difficult to replicate. I simplified all the interactions. Like, it doesn't show like the G protein binding with the A MSH. And then this is just a simplified mutation, like simplified representation. My model doesn't highlight the mutation, like it can't truly demonstrate like the changes in the protein folding and like the stability and signal efficiency in real time. But that is my presentation of MC1R protein. Thank you.